Vanakkam friends, Trinity Cultural Academy Coimbatore and New India Publishing Agency, New Delhi welcomes all of you for my presentation. And this presentation I will be dealing on the different techniques that are being used for hybrid seed production, and this also comes under the plant breeding subject. And mostly this presentation is highly useful for all competitive exams like All India Entrance Examination, Junior Research Fellow, Senior Research Fellow, and NET, Agriculture Research Service, and Senior Technical Officers, and as well as the agricultural field officers in the Institute of Banking Personnel Selection. And this is being prepared by Vanangamudi. He was the formerly Dean Agriculture and also Professor and Head of the Department of Seed Science and Technology, Coimbatore. And in this presentation, the mostly techniques and methods that are all to be used for hybrid seed production. There are three major methods. One is the single line breeding as well as two line breeding. And in this presentation, I will be concentrating only these two aspects, and three line breeding will be dealt in the next video. And as far as single line breeding is concerned, it is one parent in which the hybrids have been developed, and subsequently in subsequent generation it will be multiplied by asexual means using stolons or rhizomes or stem cuttings. The best example, classical example for this single line breeding is the NB21 grass, around Napier Bajra grass. It is a cross between Napier grass and Bajra, that is a pearl millet. Subsequently, after this hybrids, F1 will be multiplied by vegetative means, that is a slips, and that will be given to the farmers or stakeholders. As far as two-line breeding is concerned, here the two parents are involved, and when compared to three-line breeding, and this is purely both A line and B line are fertile parent. The A line and B line are fertile parent, whereas there in the three line breeding, A line is always the sterile parent. And here A line is used as female parent and the B line is used for male parent. And in this two line breeding, there are different techniques, methods, and we will see one by one. First, we will see the hand emasculation and pollination. What do you mean by emasculation? It refers to the removal of male reproductive part, that is nothing but androecium. Female parent, you should not do this in the male parent because which gives only the pollen, and this should be done in the female parent. The best examples, this technique is used in the cotton, tomato and brinjal. Now here we will take a typical example of cotton, in which how the female flowers are emasculated and then pollinated. And here, while the selection of flower buds for emasculation, you have to select a flower bud that will be opened next day, and that should be emasculated. The best time for this emasculation is after 1 p.m. and then the emasculated flowers are covered with red paper bags to prevent genetic contamination. Yeah, if there is any hybrid cotton field, if the red paper bags are there, that indicates only the emasculated flowers and that field. And regarding the before emasculation, we should understand the floral biology of cotton. We will be concentrating only on this. This is the reproductive part of the organ. This is petal, and this is the androsium, the anthers, and this has to be removed and leaving the ovary, style and stigma. This male part should be removed completely. That is the emasculation techniques, and you have to select the flower buds. That is more important. You should not select this one as well as a completely opened flower, and this is the correct stage of flower bud you have to select, and this flower bud only it will be opening next day, and in this flower bud only you have to do emasculation. And now let us see the emasculation, hand emasculation techniques. As I told earlier, you have selected the bud that is going to be opened tomorrow, and carefully by using your nail hand, by using nail, you have to remove the androsium, the anther and the completely you have to remove, and after removal you can see the flower like this. This is the emasculated flower, and this is the ovary, and this is the style, and this is the stigma. And after emasculation you have to cover the emasculated flowers with red paper bag, by maybe butter paper bag. And then this figure shows that after emasculation the flower, the emasculated flower looks like this, and next day morning you have to do hand pollination, and for this you have to select the male flowers and then collect, and that male flower pollen has to be dusted, pollen grains has to be dusted over the stigma of emasculated flower buds of the female parent. Okay, the male flowers he has to collect only from the male parent, and that has to be dusted on the emasculated flowers, particularly on the stigma of the female parent. Again, the pollinated flowers should be covered with white butter paper bags, and this shows how the hand pollination can be done. And this is the male flower which has been collected from the male parent. This is already emasculated flowers that's located in the female parent. Then after this is the pollen grain. After dusting, the pollen grains are attached to the stigma, and after pollination it has to be covered with white butter paper bags, and this is the well developed bowl after emasculation as well as pollination. This is the F1 hybrid. This is the F1 hybrid bowl. And other techniques that we will be seeing for the hand emasculation, and there are about 5 techniques other than hand emasculation. We have one suction method. This is nothing but removal of anther using thin rubber band or glass tube or rubber tube that should be attached with a suction hose. So it will suck all the pollens that is present in the anthers of the female parent. And the next technique, the hot water emasculation, and this is given to before the anther descends as well as prior to the opening flower. In the case of sorghum, it has to be exposed to 42 to 48 degrees Celsius to sterile, to make the pollen grain sterile. and in rice the temperature, optimum temperature is 40 to 44 degrees Celsius. Then the third one is alcohol treatment, that flowers, the matured flower buds will be immersed in the alcohol, then it should be rinsed with cold water, that will also kill the fertile pollen. And the fourth one is cold water treatment. It kills pollen without damaging the gynecium. And this is especially followed in rice 0 to 6 degrees Celsius. And the last one, the fifth one, genetic emasculation. You have to eliminate the necessity of emasculation by genetically manipulate the parents. And the examples I have given, the cotton hybrids examples. There are two types, interspecific hybrid and intraspecific hybrid. And we have about three interspecific hybrids, that is Varalaxmi. It is a cross between Laxmi, which is Gossypium hirsutum, and that is used as female parent, and SB289 is the male parent, which is Gossypium barbadense. Similarly, DCH32 female parent is Jayalakshmi, male parent, the hybrid is Jayalakshmi, or DCH32 the female parent is DS28 and male parent is SB 420. It is a cross between again Hirsutum and Barbadense. And one more hybrid in interspecific hybrid cotton, TCHB213. The female parent is TCH1218 and male parent is TCB209. And while coming to the intraspecific hybrid, we have two hybrids, that is Suguna and Savida, and the parent of Savida is T7 female, M12 is the male parent. Next technique, so far we have seen the hand emasculation and pollination, especially in the groundnut. And the next one, we will be seeing about the detasseling, and this is mainly being followed in maize crop. And here the maize is a monoecious crop, and the male flower, that is the tassel, that is present in the top of the plant, and the cob, which is present in the stem and below the tassel. And this tassel has to be removed, that from the female parent, that is more important. You should not remove the tassel from the male parent, because male parent only it will supply pollen to the detasseled female parent. And as I told, it is a monoecious in nature. The male flower is tassel, which is present on the top of the plant. Female flowers are spadix, which is present in the middle part of the plant. And detasseling is done when the tassel emerged out of the boot leaf stage, but before anthers have shed. This is more important. And the anthers that will be available for 2 to 4 days to days, and after complete emergence. Before that you have to remove the male tassel, and detasseling should be done every day from the emergence of the tassel up to 14 days. And this is the correct stage, the proper stage of the detasseling. And these are the completely opened flowers. This is not done for detasseling. You can see here it's a very just emerging from the boot leaf, and this is the correct stage for detasseling. Now we can see the method of detasseling, and here I have explained. Just you hold the stem. You hold the stem below the boot leaf. You have to hold the stem below the boot leaf in left hand by using your left hand, and at the base of the basal in right hand, and base of the tassel in right hand and pull it out in a single pull. This is the tassel, and by using the right hand you have to hold in the basal of the tassel and you have to pull out, single pull out, and there should not be any leftover in that, and grasp the entire tassel so that the pollen parts are fully removed. Do not break or remove the leaves, and this here the detasseling techniques is also being explained in this photograph. And after the removal of the tassel, you should not kept in the hybrid seed production plot itself. It has to be buried in the soil. If don't bury that, it will also shed the pollen and it will contaminate, it will give genetic contamination, and that will create a lot of problem. You have to take some precautions that is to be adopted during tasseling. No part should be left on the plant as it causes genetic contamination. Should be done daily. Do not break the top leaves. As removal of one top leaf, there is a loss of yield by 1.5%, and two leaves if you remove or break, and there is a loss of 5.9%. If you damage or break three leaves, there is a maximum 14% yield loss will be there. And you have to do the detasseling only in the female parent. For that only you have to mark the male parent with marker to avoid mistake in detasseling. Avoid shedding tassels, that is more important. I will be explaining shedding tassel about that in the subsequent talk, and this will cause genetic contamination. After pulling, the tassel should be buried in soil to avoid contamination. Do not practice improper, immature, and incomplete tasseling. Some of the terminologies we can see. Improper tasseling means a portion of the tassel is remaining in the plant while detasseling. That should be avoided. Immature detasseling should also be avoided because it will give lot of problem, and there is a yield loss also. Incomplete tasseling, and there should not be any remnants of the tassel at the lower portion of the things, and that should be removed. Shedding tassel means if there is in the remaining part of the tassel in the female parent, that will give some shedding pollen which may cause genetic contamination that will affect the genetic purity of a crop. And here in the maize there are different types of hybrids are there. I will be explaining one by one. Single cross hybrid, it's a cross between two inbred lines. A is inbred, that is used as female. B is another inbred, that is used as a male parent, that is a restorer line. That means it restore the vigor of the female parent, and detasseling should be done only in the female parent, not in the male parent. If you cross A and B, you can get F1 hybrid, that is a single cross hybrid. Below I have explained diagrammatically. This is the male parent, female parent, male parent. Detasseling should be done in the male parent, and the pollen will be pollinating from the male parent by either by natural pollination or open pollination. This is the hybrid seed. And examples for single cross hybrid: CoH1, CoH2, and CoHM5. CoH means Coimbatore hybrid. CoHM means Coimbatore maize hybrid 5. I have given the parents. You can please go through that. Parents also, they will be asking in the competitive examinations. And now we will see the second type, double cross hybrid. It's a cross between two single crosses. This is one single cross and this is another single cross. These are obtained by crossing two inbred lines, and in these two single crosses the detasseling should be done only in the female parent. And if you cross the one single cross with another single cross F1 hybrid, you have to do the detasseling in this female parent. And if you cross these two one, you can get the double cross hybrid. And this is again the diagrammatic representation, one single cross, another single cross. And if you cross two single crosses, you can get the double cross hybrid. Examples: Ganga 2, Ganga 101, and CoH3. And these are all the parents involved in double cross hybrid. Please kindly go through this one. And the third one is three-way cross. It is nothing but crossing between single cross hybrid and an inbred line. That is the three-way cross. Don't confuse. There may be a top cross, double top cross, and here let us be very clear that and one single cross hybrid will be pollinated, crossed with the inbred line, and you can get the three-way cross. Examples: Ganga 5, CoHM 4. And the fourth one is a double top cross. It is a crossing between single cross and open pollinated variety. That's a popular variety of maize that will excel with other varieties or hybrid. That has to be crossed with the one single cross hybrid and open pollinated variety, and you can get the double top cross hybrid. Whatever may be the things, you have to do detasseling in the female parent. Okay, and the last one is top cross hybrid. It is a hybrid produced by crossing of an inbred line with a certified open pollinated variety. Here you have to do detasseling, and if you cross inbred with open pollinated variety, you can get top cross hybrid. Now in this table we can see the different stages, maybe the classes of seed production, how it is being produced in different hybrids in maize. And single cross, the breeder seed, A is multiplied separately, B is multiplied separately. Like this in double cross, A, B, C, D parent has to be multiplied for production of breeder seed. In three-way cross, A, B, C, three inbreds are multiplied, maintained for breeder seed. In double top cross, two inbreds and one open pollinated variety should be raised separately, and you have to multiply the breeder seed. And top cross, one inbred and another OPV. All the inbreds and open pollinated variety, by keeping the isolation distance, you have to multiply breeder seed. That breeder seed will be given for the production of foundation seed. Here also the A and B in separate blocks or separate bits, keeping isolation, you have to raise. Here in double cross, one single cross hybrid and another single cross hybrids are raised separately, and while doing that we have to detassel in the female parent like A and as well as B. And three-way cross, A and B and C has to be multiplied in larger quantities. And in double cross, A and B and OPV has to be multiplied in different fields by keeping isolation. Here also A inbred line, OPV is multiplied in different fields. In order to increase the quantity of foundation seed that will be supplied for seed production. Okay, certified seed production. Here A and B is raised in the same field by keeping the what do you mean by that is the planting ratio either 4:2 or 6:2. That will vary with the crops. If it is maize, the 4:2 and 62. If it is rice, 8:1 or whatever it may be, that we can see separately in the things. And here also the double cross A and B is a female parent that should be raised along with the C and D by using the planting ratio. Here A and B and C is raised. A and B is a single cross, that is a female parent, and C is the male parent that has to be raised in the uh, field hybrid seed production field, adopting the planting ratio. Similarly in double top cross as well as top cross. I think with this, I will be closing this presentation, and regarding the 3-line breeding, we will be seeing in the next video. Kindly watch this video and share this presentation, share this video to your friends, like and comment. Comment is important to improve our presentation, and kindly subscribe the channel. Thank you very much.